Genomes. Let us have an overview of the genome anatomies. Biologists recognize that the living world comprises two types of organisms. One, the eukaryotes, which comprise of animals, plants, fungi and protozoa, whose cells contain membrane bound compartments including a nucleus and organelles such as mitochondria, and the case of plant cells, chloroplasts. The second group, prokaryotes, whose cells lack extensive internal compartments. Prokaryotes are distinguished from one another by the characteristic genetic and biochemical features, and they are further grouped into two: the bacteria, the commonly encountered prokaryotes, the gram negatives, for example Escherichia coli, the gram positives, example Bacillus subtilis, or the cyanobacteria, example Anabaena, and many more. The archaea, which are less well studied and have mostly been found in extreme environments such as brine pools, hot springs and anaerobic lake bottoms. How many types of genomes exist? A genome is the genetic material of an organism. It consists of deoxyribonucleic acid or DNA and RNA, ribonucleic acid in RNA viruses. There are 5 types of genomes: prokaryotic genomes, eukaryotic genomes, nuclear genomes, mitochondrial genomes and chloroplast genomes. Prokaryotic genomes. Prokaryotic genomes are very different from eukaryotic ones. There is some overlap in size between the smallest eukaryotic genome and the largest prokaryotic genomes, but the whole prokaryotic genomes are much smaller. For example, the genome size of Escherichia coli K12 is 4639 kilobase pairs, 2/5 of the size of the yeast genome. In prokaryotes and eukaryotes, the physical organization of the genome is also different. The entire prokaryotic genome is contained in a single circular DNA molecule. Other than this single chromosome, prokaryotes may also have additional genes on independent smaller circular or linear DNA molecules called plasmids, as shown in figure 3. Genes that are carried by plasmids codes for properties such as antibiotic resistance or the ability to utilize complex compounds such as toluene as a carbon source. A prokaryote can exist quite effectively without plasmid. The prokaryotes display a considerable diversity in genome organization, some having a unipartite genome, which means they are not divided into segments like Escherichia coli, but others being more complex. For example, Borrelia burgdorferi B31 has a linear chromosome of 911 kilobase pairs, approximately 853 genes, accompanied by 17 or 18 linear and circular molecules, which together contribute another 533 kilobase pairs and at least 430 genes. Bipartite genomes, the genome segmented into two segments referred to as DNA A and DNA B, are now known in many other bacteria. In one respect, E. coli is fairly typical of other prokaryotes. The prokaryotic genomes are even more compact than yeast and other lower eukaryotic genomes, as shown in figure 4a, which shows a 50 kb segment of Escherichia coli K12 genome. There are more genes, almost 43 genes, and less space, 85.9%, between them. Some genes have virtually no space between them. For example, in the case of synthesis of the amino acid threonine, the genes THR-A and THR-B are separated by a single nucleotide, and THR-C begins at the nucleotide immediately following the last nucleotide of THR-B. These 3 genes are an example of an operon, a group of genes involved in a single biochemical pathway. Operons have been used as model systems for understanding how gene expression is regulated. In general, prokaryotic genes are shorter than their eukaryotic counterparts. The average length of a bacterial gene being about 2/3 of an eukaryotic gene, even after the introns have been removed from the latter. Bacterial genes appear to be slightly longer than archaeal ones. Two other features of prokaryotic genomes can be deduced from the figure 4e. Firstly, there are no introns in the genes present in this segment of the E. coli genome. In fact, E. coli has no discontinuous genes at all, and it is generally believed that this type of gene structure is virtually absent in prokaryotes. Of course, a few exceptions occurring mainly among the archaea group. The second feature is the infrequency of repetitive sequences. Most prokaryotic genomes do not have anything equivalent to the high copy number genome-wide repeat families that are found in eukaryotic genomes. They do, however, possess certain sequences that might be repeated elsewhere in the genome. Examples being the insertion sequences IS1 and IS186 that can be seen in the 50 kb segment as seen in the figure 4e. These are further examples of transposable elements, the sequences that have the ability to move around the genome, and in the case of insertion elements to transfer from one organism to another, even sometimes between two different species. The figure 4 shows a comparison of the genomes of humans, yeast, fruit flies, maize and Escherichia coli. A shows the 50 kb segment of the human beta T cell receptor locus compared with the 50 kb segments of the genomes of Saccharomyces cerevisiae, chromosomes 3, and C shows the Drosophila melanogaster, D is that of maize, and E. coli K12.